So welcome to the second series, and this second series is about the Harrison rapid revision in 8 minutes. If you like the content, you have to share and subscribe, please. As I told that Harrison's was directly quoted from NEETSS, so this is my second topic that I'm uploading today. I have many topics. If I receive your support, definitely I'm going to upload more and will help you ace the NEETSS. And you should not worry about the first 400 and the last 400 pages of Harrison, which are very important, and 50 questions might be asked from that. So starting with this, it's a mitochondria. It's a cell organelle for oxidative phosphorylation. What you have to do is you have to either take your Harrison or you have to just note down the points which I'm telling. 10 minutes, one topic, over. Don't go back to Harrison again. Okay, so it's responsible for the electron transport chain, coenzyme complex 2, complexes 1 to 5, and cytochrome C. They are located in the inner mitochondrial membrane. So they are directly asked line by line from Harrison, including complex 1, 5, like that. So just don't think that it's not going to be asked. Whatever I'm telling is 100% the potential questions. So mitochondria, they are the predominant source for reactive oxygen species, ATP production also, right? For functions, for all cells and tissues, and tissue or cell specific. They can always give certain things, right? When it comes to a spell specific, like all the cells and tissues, 4 functions are there, like oxidative phosphorylation, free radical formation, calcium homeostasis, and apoptosis. But only in certain tissue or cell specific. There is metabolism. This is important. Hepatic ammonia detoxification, neurotransmitter metabolism. So predominantly, if it is tissue or cell specific, it is mostly metabolism. But if it is all cells and tissues, you should always tell there are only 4, including the calcium homeostasis, apoptosis, free radical production, oxidative phosphorylation, right? This is a very important table telling the functions. Next, coming to the mitochondria. The mitochondria has itself a own DNA. It's called mitochondrial DNA. It does not require chromosomal DNA, right? So the inheritance. If there is a mitochondrial DNA, it is always from the mother. So all offsprings are affected, right? Next, we have this mitochondrial DNA here, which Harrison has marked in red color. If there is a dual genetic control and multiple organ systems involved, because mitochondria is there in all the cells, almost all cells. Pearson syndrome, if it affects the blood, neutropenia. Pancreas, there is diabetes mellitus. Kidney, Fanconi syndrome and glomerulopathy. Liver, hepatopathy. Eyes, optic neuropathy, ophthalmoplegia, retinopathy. Heart, it involves the conduction disorders, Wolff Parkinson-White syndrome, cardiomyopathy. Skeletal muscles in the form of myopathy, neuropathy, weakness and fatigue. Brain in the form of seizures, myoclonus, ataxia, stroke, dementia, migraine. Nuclear DNA is always separate, okay? It's separate. That is what this arrow is telling and the pointer is telling. Mitochondria always secretes ATP. If ATP is gone, all this entire complex occurs, right? This is what you want to know from this table. Okay, and change the color now. This POLG gene is also being asked. The mutations in the POLG gene is also very important. It's a mitochondrial DNA gene. It affects the polymerase function of the mitochondrial DNA polymerases. Right? So I told you it's a maternal inheritance. Mother is affected. All the offerings are affected. Mother is affected. All the offerings are affected. This one is not married, so obviously no offerings. Father is affected. Offsprings are normal. Mother is affected, gone. Father is affected, normal. So this is also very important, right? The next, see, there are various syndromes. So what I have told you here is an array of syndromes. Then these entire syndromes will be discussed in this table, which is right here in the Addisons, right? You just don't have to worry about anything, all those things, and just study whatever I'm going to tell you. Yes. So a dark color would suffice. Yes. If it's a Pearson syndrome, there is pancreatic insufficiency causing diabetes, pancytopenia, lactic acidosis. So lactic acidosis is common. This is MELAS. This is mitochondrial ME encephalopathy LA. Lactic acidosis S. Stroke-like episodes may manifest only as diabetes mellitus. This is very important. Next, MRF. MRF, myoclonic epileptic red ragged fibers. It's in the muscle, right? Because of the muscle involvement, there will be ataxia as well as a nerve involvement. In addition to that, there will be a sensor neuron hearing loss and there will be dementia, which as increased CSF protein, it is MRF, right? Next, deafness is also very common. It can be a isolated non-syndromic sensory neural hearing loss, or it can be a progressive hearing loss. And these mitochondrial disorders, they are increased susceptibility to aminoglycosides, which is given here, right? Next, there is a chronic progressive external ophthalmoplegia. Okay. Next is Keene-Sars syndrome. This is very important. It is also associated with external ophthalmoplegia, ataxia, heart block, and retinal pigmentation. The next is Lake syndrome, which is also called as NARP. That is associated with loss of central vision leading to blindness. So Lake syndrome is blindness. Keene-Sars is ophthalmoplegia. CPEO is ophthalmoplegia, right? And in this table, what is very important is that where is homoplasmy is these two. Homoplasmy is the same presentation. Heteroplasmy is the different presentations of the mutations. So remember, what are homoplasmies? It's only deafness. Rest all are heteroplasmy. Okay. And in this table, one more thing that you have to remember is sometimes there might be a somatic mutations. These are CPEO, Keene-Sars syndrome, and Pearson syndrome. Keene-Sars also has a progressive external ophthalmoplegia. So you can remember these three. This is what they're going to ask. And one more thing is which is associated with a very large deletions. That is Pearson syndrome, which is associated with a very single deletions are CPEO. And a 5 kb mutations is Keene-Sars syndrome. These three also you will have to note down. Single deletions is CPEO, large deletions is Pearson, 5 kb deletions, which is a common deletion, is Keene-Sars syndrome, right? This is what you have to remember. And they might give this diagram as well, like mitochondrial encephalopathy. Like they can give a question and they can ask you, right? And Lake syndrome is Na nucleotide. It is called as, it's associated with usually ataxia too, right? It is associated with ataxia. Just don't worry too much about this. There is only thing that there is degeneration that is involved in cases of mitochondrial disorders. Okay. Whenever you're going to test, you have to test for CK, various organs, wherever it is involved. Whenever you suspect, you should always take lactose. Diabetes is also very common manifestations. CNS involvement, suspecting cardiomyopathies, cardiac, brain also, and EMG should be done. And also point specific mutations, which I have already told you. Muscle biopsy, red rag fibers, yes. And sometimes they might do a enzymology, complex 1 to complex 5, which I've already told there, right? This is all that you have to think when it is a mitochondrial disorder. Don't worry, it is associated with apoptosis as well. This is involvement, Paul gene involving the polymerase, and always you have to do a genetic counseling, because it's 100% disease if the mother is having, right? And there is